Hello, this is Fred Tully from Mibelle Biochemistry, and I would like to welcome you all to our webinar on our new launch of a new product called Snow Algae Powder. Now, this is a new product which is based on what we call the secret of red snow. And I want to introduce to you to Daniel Schmid. He is our head of research, and he will tell you more about what is the secret of red snow. Daniel, please.
So first, hello from my side. So what is the mystery of red snow? In summer, persisting snow fields on high mountains sometimes show a red-pink color. The color comes from spores of snow algae. The red color protects the algae against extreme UV radiation. There are many different species of snow algae known. Snow algae have a life cycle with two phases. In spring, when the snow starts to melt, the spores change into green organisms that move with flagella to the snow surface, where they use the light to produce energy. The snow algae start then to propagate. In early summer, when the UV light is strong, the algae change back to red spores. The red color comes from carotenoids, compounds that protect the cells against UV. In the form of spores, the snow algae survive under a blanket of snow until next spring. Snow algae are extremophile species adapted to live under extreme conditions. Extreme temperature, strong UV radiation, lack of nutrients. They live on water, CO2, sunlight and minerals. Their growth rate is highest in spring at temperatures between 0 and 5 degrees. To survive in such an extreme habitat, snow algae have developed a series of adaptations that are reflected by diverse secondary metabolites. Green chlorophyll is used to transform light into energy, a process called photosynthesis. The red stage is the dormant phase where the following metabolites are present. Carotenoids, these are fat-soluble antioxidants, water-soluble antioxidants, many different polyphenols, biopolymers, antifreeze glycoproteins, and osmotically active amino acids and sugars. These are all interesting metabolites with potential anti-aging effects in human cells. To use snow algae as raw material for a cosmetic ingredient, we had to develop a biotechnological production system. The first step was harvesting of snow algae in the mountains. The cosmetic ingredient, the snow algae powder, was produced the following way. In a tube photobioreactor, favorable conditions were used to stimulate propagation to produce biomass. This is the green phase. Then the conditions are changed, nutrients are reduced and light strongly increased to force the algae to produce carotenoids and the other interesting metabolites. This is the red phase. Then the red spores are harvested, the cells are disrupted by high pressure homogenization, and fragments and cell content are incorporated into liposomes. Finally, the liposome solution is dried on a maltodextrin carrier material. This is the exact composition of the snow algae powder. Snow algae extract, maltodextrin, phospholipids, and water. The INCI for the snow algae extract is not yet confirmed.
So we have here a very nice new source to make a fantastic new cosmetic raw material, and it's produced in a sustainable way by biotechnology. Now, further to that, we have recognized or discovered a new anti-aging activity with this snow algae powder, and it's called caloric restriction mimetic anti-aging activity. So what is this? Caloric restriction is nothing new. It was already discovered in 1934 when Crowell and Clive Mackay observed that the reduction of the caloric intake in mice led to an increase of their life expectation. So we see here in the slide that the average life from a mouse from about 30 months could be extended to something like 55 months. So the reduction of calories led to an extension of life expectation. This could also be repeated in mice. Now, the secret of long life by caloric restriction could also be confirmed in yeast, worms, mice, rats, dogs, apes, and it's also on a good way in studies with men. So food limitation to increase lifespan could already be observed in the world wars. So the reduction of caloric intake actually led to an extension of life expectation. And of course, it's also well known that fasting cures, as practiced by different religions, lead to a health benefits. Today, a 5-2 diet is very popular. This means that you eat on 5 days and you fast on 2 days, and this not only controls your weight, but also adds health benefits. In Okinawa, there are some residents which have a very low energy diet, and there they observed a large number of centenarians. So all these data show that caloric restriction really shows some interesting health benefits and life extensions and anti-aging effects. So how does this work on a biochemical way? So we know at a high nutrient level there's a lot of insulin. Insulin will bind to the insulin-like growth factor receptor shown on the left side of the slide, which after a few further steps will deactivate a transcription factor called FOXO. In a low nutrient level, that means there is no or less insulin present, this insulin-like growth factor receptor is active and actually activates a transcription factor FOXO, which will activate anti-aging genes like DNA repair genes and detoxification genes, and by that leading to longevity. Exercise in excess to low nutrient levels can also have a positive influence on this FOXO transcription factor through a gene called AMPK. Now there are two factors which stimulate longevity pathways. At a high nutrient level, that means there's a lot of insulin present, there is a gene called CLOTO which can still activate FOXO even though the insulin-like growth factor receptor is blocked or is bound to with insulin. And the other factor is AMPK, who can further activate FOXO to lead to longevity. So what is the CLOTO gene? The CLOTO gene was discovered in 1997 and was a gene causing extreme aging when this gene was underexpressed. The gene was named after the Greek goddess CLOTO. And if CLOTO is high, then also FOXO is high, and this means an anti-aging effect. The other interesting protein or gene is AMPK. It's a master regulator of metabolism. It's activated when energy is low. That means, for example, at caloric restriction conditions, but also it is activated upon exercise. So an AMPK actively declines when we age and upon nutritional overload. So that causes increased oxidative stress. Now, that these are really important genes for lifespan is known by some mutations, and I will show you some mutations for longevity which are well known and researched. So mutations in the insulin-like growth factor receptor are known for horses and dogs, and it's known that smaller dogs and smaller horses always live longer than the big dogs and the big horses, based on specific mutations in the insulin-like growth factor receptor. There are also some Japanese cohorts known for longevity, and also there the insulin-like growth factor receptor 1 mutations could be identified as the reason for this longevity. Regarding FOXO mutations, there are 8 independent cohorts known around the world which show longevity and long-lived subjects. So this clearly demonstrates that these FOXO and insulin-like growth factor are very important genes for longevity. Now, that this has some impact also for the skin was discovered in 2009 by a Japanese research group. They could show that UVA and UVB radiation actually will downregulate FOXO1, and this will then decrease the collagen synthesis. This will increase MMP expression, and of course we all know that this will cause wrinkles and photoaging. So this means wrinkles and photoaging are actually also regulated by the transcription factor FOXO1. Now we have seen that caloric restriction and exercise are key factors for lifespan and health span. So, and the goal now is to search a mimic activity which will mimic the caloric restriction effect. And this would cause the extension of health span and lifespan on skin cells without diet and without exercise. And for that, we searched for an activity stimulating Klotho and AMPK, the two main targets of this caloric restriction mimetic pathway. With the next few slides, we are going to show you some of the studies we have done to show the activity of our snow algae powder in the case or in the direction of caloric restriction mimetic activity. And Daniel will guide you through these studies.
In cell culture assays, we could demonstrate stimulation of the expression of the Klotho gene and of AMPK activity by the snow algae extract. In the first assay, we used fibroblast cells and cultured them over several weeks. The cells are transferred every week into fresh medium. This is called a passage. The cells start then to proliferate again. Cells cultured over 17 passages show signs of aging and can be regarded as old. Expression of the Klotho gene was measured by quantitative PCR. Here is the result of the Klotho expression analysis. In aged fibroblasts, the expression of the Klotho gene was clearly reduced compared to young cells. But when aged fibroblasts were treated with the snow algae extract, Klotho expression was strongly stimulated up to a level even higher than in young cells. Activation of AMPK was demonstrated on human keratinocytes. The cells were cultured in presence of the snow algae extract and insulin imposing a calorie stress conditions. AMPK and phosphorylated AMPK, which is the activated form, were analyzed with Western blot. Here are the results. The ratio of activated versus total AMPK increased in presence of the snow algae extract at 1% under normal conditions. But under high high insulin conditions in the calorie excess medium, the increase was more pronounced, accounting to over 100%. Thus, the snow algae extract stimulates the phosphorylation, means activation of AMPK in a dose-dependent manner. It is a protective effect against caloric excess induced by high insulin concentration. It prevents the age-related decline of AMPK. It is an activation of the longevity pathway. We also used the aged fibroblast model to analyze collagen production. Aged fibroblasts at passage 17 were cultured with and without 0.1% snow algae extract. Quantitative PCR was used to analyze the expression of collagen 1 and 3. Both collagen 1 and 3 were downregulated in aged cells. Incubation with the snow algae extract reduced the downregulation of collagen 1 and strongly stimulated the expression of collagen 3. Thus, the snow algae extract helps to repair the effects of aging by stimulating the most abundant collagens in the skin. The extract was also tested in an H2O2-induced senescence model. Fibroblast cells were treated with the extract and then exposed to H2O2 to induce senescence. Quantitative PCR analysis was used to measure expression of 64 aging genes. We found an anti-MMP effect. MMP stands for matrix metalloproteinases, enzymes which degrade collagens. Gene expression analysis showed that the matrix metalloproteinases 1 and 3 were highly upregulated after H2O2 stress. But in cells pre-treated with the snow algae extract, the upregulation was much lower. The snow algae extract counteracts the effects of aging by reducing the MMP activity in senescent cells. We also did proteomics to study the effect of the snow algae extract. Gene expression analysis is not always reflecting the real impact because it measures formation of mRNA, which is only an intermediate between a gene and its product, the protein. Proteomics is the analysis of the concentration of a series of different proteins and enzymes. In this study, we measured the level of more than 100 proteins in primary human keratinocytes. The cells were cultured in a pro-aging medium in presence or not of the snow algae extract. The pro-aging medium lacks growth factors and protecting compounds. This figure shows the effect of the pro-aging medium. The proteins are ordered in categories such as adhesion, antioxidant activity, DNA repair, electron transport, metabolism, protein synthesis, and stress responses. Compared to a normal, rich medium, the levels of the analyzed proteins changed in average by up to 40%. The levels of all categories could be normalized with the snow algae extract. Enzymes associated with protein synthesis, metabolism, DNA repair, and electron transport were less reduced, and enzymes associated with stress response, oxidized protein turnover, and redox antioxidant mechanisms were less increased after incubation with the snow algae extract. I would like to go over now to clinical studies. We wanted to analyze the effect of the snow algae powder on the dermal-epidermal junction. It is the area that tightly binds the dermis to the epidermis. Its permeability regulates exchanges between both tissues. In young skin, the structure of the dermal-epidermal junction is wavy, maximizing the surface area of connection between the epidermis and the dermis. In aged skin, the dermal-epidermal junction becomes more flattened. The connection between the epidermis and the dermis is reduced. The two-photon microscopy was used to analyze the dermal-epidermal junction. It is a non-invasive 3D technique to analyze structures in the upper dermis. An infrared laser generates autofluorescence from elastin and a second harmonic generation signal from collagen. After scanning of the skin at different depths, 3D pictures of the dermal-epidermal junction as shown here can be generated. A gel with 2% snow algae powder was tested on several volunteers over 2 months on the inner side of the forearms. After the treatment, the dermal-epidermal junction was improved, shown in this picture, in this vertical scan. The two-photon microscopy data could be used to calculate the surface of the dermal-epidermal junction, and compared to the placebo, the surface could be increased by 30.5%. In another clinical study, we could demonstrate a reinforcement of the skin barrier in challenging conditions. It was a placebo-controlled half-site study with 20 volunteers over 3 weeks with alpine winter climate. The volunteers spent much of the time outside where the skin was challenged. An emulsion with 3% snow algae powder was applied on one half of the face and the corresponding placebo on the other half. Transepidermal water loss and age spots were analyzed. The winter weather conditions led to an increased loss of water by the skin. In the placebo-treated half phase, TEVL was increased. Snow algae powder was shown to counteract this effect and even to reduce TEVL. The study showed also a reduction of the visibility of age spots. High-definition photographs were taken using VisioFace Quick. With the VisioFace system, it is possible to lighten the skin in a way that pigment spots gets easily visible. After 3 weeks, there was an improvement in the snow algae-treated face, whereas on the placebo side, a worsening in the spots was found. With snow algae powder, the visibility of age spots was reduced in 76% of the cases. Another study was conducted to show a smoothing and moisturizing effect. An emulsion with 2% snow algae powder was tested on 20 women over 4 weeks. There was a clear anti-wrinkle effect demonstrated here with 3D before and after pictures, and skin hydration was improved by 10% compared to placebo and by even 25% compared to untreated.
Thank you, Daniel, for this explanation on the studies. So we have a new product from a very exciting source which follows a new anti-aging mechanism by caloric restriction mimetic activity. And based on this product, we think that a few interesting new claims can be made in a cosmetic formulation with such a product. So here we want to show you some claim ideas for this product. We can say snow algae powder protects and activates the longevity factors in the skin cells. It rejuvenates and protects skin off cellular level. It safeguards the skin's usefulness by activating collagene, a new gene in the cosmetic field. It will strengthen cellular defense mechanisms through caloric restriction mimetic activity. Again, this is a new claim in the cosmetic field, caloric restriction mimetic activity, and the product will reinforce and smooth and hydrate the skin. So the application of such a product are all type of rejuvenating and repair formulas, age defense products, youth protecting and promoting skincare formulas, formulations to increase the skin's longevity, face, body, and hand. So there are a number of marketing benefits for this product. It is based on a unique new extremophile algae coming from the Alps. It is a sustainable production by biotechnology. It is a novel anti-aging concept by calorie restriction mimetic activity. And we have created a snow algae logo which can be used on finished product using this material. There is also a website on snow algae available. The product as such is preservative-free, in powder form, and we have already received a granted patent in the United States. So with this, we come to the end of this webinar. If you have questions to this product, you can email us your questions, or more easy, just visit us at InCosmetics at our stand in Hamburg at stand 1G50. And we all thank you very much for joining our webinar and hope to see you in Hamburg. Goodbye.